think we'll go ahead and move on at this point and talk about using population-based animal models. And Jeff French from the NTP will be our next speaker.
I'm going to try to go back to the podium.
Okay.
Getting a lot of... And this will turn yellow.
I want to thank the Academy and the board, especially the organizers, for making this, and the sponsor for making this meeting possible. To follow up on what the previous speakers and the points they have made, especially what Fred has just said, I'm going to tell you now about two different models, which we will call a population-based mouse models, that will help inform, I believe, human studies as well. Okay, so to put this in context, you're all familiar with this already. Basically, we've relied historically on both epidemiology as well as animal models of human disease to identify and characterize hazard and to provide quantitative data for risk assessment. Often, epidemiologists have observed the relationship first, and which provides strong support for identifying an agent as a carcinogen. But animal models have also played a critical role in this, and often have identified a candidate as a carcinogen prior to human exposures being recognized. There are some weaknesses in both of these processes, though. Lack of, often with good exposure assessment data on both environmental and occupational exposures, and the role that both inter- and intra-individual variation plays. The disease endpoints are categorical, not always quantitative, and there could be extreme ranges in individual variability depending on the test population. More recently, it was alluded to earlier, both the candidate gene association studies as well as genome-wide association studies are now playing a valuable role in this. But often the GWAS studies also lack exposure assessment data, and it makes it difficult to relate it to specific exposures. In the animal models, we have been handicapped by the limited use of a single strain of one or two species or outbred stock of those species as our test model. And we often don't know where the results fall within type 1 and type 2 eras. And there may be other approaches that we should be considering this in this context. And so today I'm going to show you data from studies with benzene, a ubiquitous carcinogen that's been well studied, and that was the reason it was selected, because there's robust data in both human exposures and animals, particularly different strains of inbred mice and rats that make this particular interesting. Now note that, as Steve Rappaport has shown us, this is a very complex metabolic scheme showing both phase 1 and phase 2 enzymes that give rise to a number of different toxic reactive intermediates, electrophiles that can act either as the muconaldehydes do, as clastogens, or as other genotoxic mechanisms. And it's been shown that knocking out these critical genes also almost always ameliorates or minimizes toxicity. So there are a number of important genes in this landscape that are critical to understand. But principally, the Lange study in Science in 2004 showed that it was difficult to ascertain no adverse effect levels for this very important compound, and that we need to try other approaches, I believe. So the first model that we started out with was a panel of inbred strains. We had recently finished sequencing 15 inbred strains with Perligen Sciences. And we added to that, that was in regard to the reference of the C57 Black 6 sequence, genome-wide sequence strain. And we added to that the B63F1 as well as the PW-LAC-J because it turned out the related strain that we had sequenced had significant introgression from laboratory-derived models and made it more difficult to make interpretation. So from this 18 inbred strains, we determined to look at ADME. It was alluded to earlier by Bill. Absorption, distribution, metabolism, and elimination, and the consequences of that in both the blood and target tissues, in this case, bone marrow. This shows you a rank order on the left of the individual inbred strains against their blood AUC in males after being exposed to 100 micrograms per kilogram of a ring label benzene. So this is the total amount of radioactivity observed over a 2-hour time period that allowed us to calculate the kinetic estimates of AUC, Cmax, Tmax, and elimination rates. Note that there's a 10-fold difference across this for the AUC. Note particularly also that the black 6 and C3 parental lines to the B63F1 had a similar phenotypes, but note the different phenotype in the B63F1, which had a much higher AUC, and might be responsible that it was a good model for the outcome in oral 2-year studies that help support benzene as a carcinogen. Note also particularly that the wild-derived lines here, the PWK, the CAS, the PWD, in terms of AUC are very similar. But when you look at, I turned out of order. I also wanted to show you how this data can be used using haplotype association procedures to actually look for causally related SNPs that are associated with this phenotype. Shown here on the bottom is the cumulative genome position from all 19 autosomes and the X chromosome on the y-axis, and the strength of the association on the x-axis, which identifies a number of potential candidate genes. And I'll point out particularly the COPA, the coatomer proteins, which are critical for packaging materials, moving them in and out of the cell, as well as recycling those for further. Also, I wanted to point out the EDNRB gene, which is an endothelium receptor, which when knocked out in mice causes changes in systolic blood pressure, which calls into question the role of physiological parameters and how these phenotypes are affected. In this case, the strength of the association is suggestive and not reaching genome-wide significance. In terms of clearance, and this was skipping directly to clearance because there were no dramatic differences between Cmax and Tmax under the conditions of this experiment. But we see that clearance shows also about a 10-fold range of difference across this panel of inbred strains. And again, the parental lines for the B6C3F1 is quite different from the F1 hybrid, which in this case has a very low clearance rate, which is associated with its high AUC. And that the PWK and the PWD, compared to the CAST-ICR-J, show differences. Note that the PWD shows an expected correlation, but the PWK line didn't change. It had both had a high AUC and a low clearance rate. Obviously, something genetically is happening there. In the haplotype association map, which is probably compromised at some level because the extent of the identity by descent in this panel of inbred strains was more robust for clearance phenotype as opposed to the AUC phenotype. And we see a number of genes that rise to genome-wide significance, including the ectonucleoside triphosphate diesterase, which when knocked out also changes systolic blood pressure. All of these become candidates that ultimately will have to be, that identified in this discovery phase, will have to be functionally validated to show causal relationship. Interestingly, when we correlate the blood AUC against the blood clearance, it shows a significant correlation. And also, the AUC, it correlates highly with the systolic blood pressure in these strains, but not with clearance. Moving on to the target tissue, bone marrow, the AUC shows a 19-fold difference across these particular strains, with significant variation between the different strains. Again, B63F1 is different from the parental lines in this case, which most likely evidence of an epistatic interaction occurring, a gene-gene interaction that somehow is not dominant, it's not recessive, it is actually an exacerbation of the phenotype. However, there was no identifiable associations within this genome for this phenotype, suggesting that in these panel of strains, there is not sufficient heterogeneity to identify a association between the genotype and the phenotype. Going to clearance, even more dramatically, there's a 36-fold difference across this panel of inbred strains. Again, showing the same feature that the B6CF1 is different from the parental lines, and that the cast is intermediate for clearance, for the clearance phenotype with the other wild-derived lines, and this will become more important when I get later on to the second model. There was a bit more robust association in this panel for clearance in the bone marrow phenotype, and principally the candidates on chromosome 9 and 15 are associated with vesiculation of proteins and monocarboxylic acids, so it may play a role in clearance in this model. But at this point, with this data, we have to assume that even though the ADME studies showed a 10 to 36-fold difference across this panel of strains for AUC and clearance in both blood and bone marrow, we often fail to achieve genome-wide significance to be able to identify associated SNPs. The Phenom Panel, which is a panel supported at JAX, is often used. It's been used by a number of investigators successfully, comprising of 35 to 40 inbred strains. We have determined to be difficult to use for both genetic and epigenetic association studies due to both the lack of genetic power as well as the inefficiency to load studies. Some of these inbred strains are very difficult to breed. It's very hard to initiate studies with a sufficient number of mice at a reasonable age at a time. But most importantly, I believe that they are somewhat compromised due to the large identity by descent in the laboratory inbred lines in particular, that leads to low accuracy in genetic analysis. Consequently, I will propose to you today that we need a better population-based model. And the one I'm going to propose, the initial studies were recently reported in Genetics, and that's the Jackson diversity outbred model. This is a new mouse resource. I see the relationship that the work done by NTP and NIHS with Perlogen back in 2003 to 2005 had a direct effect on this, because we understood that the laboratory mouse compared to the wild-derived mouse was quite different than we originally expected. It was not an admixture of different the three subspecies as believed. It was actually largely of domesticus species. It actually showed significant identity by descent. This, I believe, and you can tell me later whether you believe this might be a good model for environmental exposures, toxicity, and disease. It was developed by the complex traits community as a resource that developed from the collaborative cross. And let me explain both of these. The collaborative cross is an 8-way advanced recombinant intercross lines that was conceived by David Threadgill and colleagues 10 years ago. And they selected, the first 5 are laboratory-derived lines, A, the black 6, the 129, the NOD, the NZO, and 3 wild-derived lines inbred, CAS, PWK, and WSB. This shows the letter codes and the color codes I'll be using in subsequent slides. And I point out that in the supplementary reading materials you're provided, there's a list of references that if you're interested in this, I encourage you to read. It can give you much more background than I can this morning. Their initial target was 1,000 different recombinant inbred lines. Just like having one inbred line, they were going to create 1,000 genetically defined inbred lines. The way they did that, they chose these 8 inbred lines. They created funnels for doing multiple intercrosses. This shows funnel OR 867. It shows in the 8 parental lines, the females and males from each of the lines were intercrossed for generation 1, intercrossed again for generation 2, and then brother-sister mated for 20 generations to fix the genome in this context. This shows a representative haplotype structure of the first inbred line, CC0001 at UNC. And it's interesting that that they have been able to observe over 45 million segregating SNPs in this population. This is in the context of similar magnitude to human genetic diversity. Probably not the same genetic diversity, but genetic diversity nonetheless. At the 4th and 5th generation of this, Gary Churchill at JAX took over 200 males and females and created a founding population to create a diversity outbred stock. I want to point out that most of the outbred stocks used in toxicology today had a very small founding population, no more than 2 or 3 males or females of each. Subsequently, even though they're putatively outbred, they have limited genetic diversity and they're mostly inbred. I may get in trouble for saying this, but that's the truth. The DO mice show some very interesting features that make them very useful, I think, for determining the wide range of variability in response to a toxic exposure, and allows it to be an ideal mapping tool for this. Even at the 7th and 8th generation, the haplotype structure reveals that most of the genomic segments are 4 to 8 megabases, usually 40 to 60 genes in that size. It makes it a reasonable segment to interpret and test. Most importantly, even every mouse has either equal to or greater than 10% minor allele frequency. It's an experimental model, I'll remind you, but it helps eliminate the consequence of rare allelic variants that occur very frequently, but often have, when they are observed, have very high light scores in this context. And at the bottom, you see 2 sets of litter mates that differ in coat color, in size, etc. It's truly a remarkable new resource. To test this, we had made the shift from a panel of inbred strains to test the diversity outbred stock because we thought it would be more robust. Well, you can evaluate and tell me whether it is or not. We chose to use 1 in 10 parts per million benzene, potentially human-relevant exposures occupationally, compare it to the air control, and then test 100 parts per million because we knew that to be a carcinogenic or tumorigenic dose. So we set our high dose at a known tumorigenic dose, and then determined to use some biomarkers of DNA damage and hematotoxicity for our phenotypes. Exposure for 28 days to reach equilibrium in terms of generation of either micronucleated reticulocytes or their depression of the bone marrow in this context. The mice were from the 7th and 8th generation. They were selected from 175 breeding pairs, which is much larger than you normally get from your normal vendor. And they were randomly assigned to exposure group by weight. And we allocated nervously 75 mice per exposure group. For 4 exposure groups required us to use 300 male mice per study. Why male mice? There's demand for these, and the first study we could do was with male mice. Also, the fact that benzene seems to be more toxic to male mice. If you neuter them, they become feminine, and they don't respond to benzene toxicity the same way that the males do. I have just a few more minutes, so I want to be sure to get through this. So this shows you the range of response. Notice that at the bottom, across the y-axis, there is the 2 cohorts shown for 0, 1, 10, and 100 parts per million exposure. We've looked at micronucleated reticulocytes as an estimate of DNA damage, and remarkably, it shows that there are a large number of individuals that have significant DNA damage, but there is a population that do not respond to a carcinogenic dose. These are about a third of the mice in this population. 2 different studies, 75 mice per exposure group at 100 parts per million. We're seeing no differences. We pool those together for doing linkage analysis. Linkage analysis revealed 2 very strong QTLs. On chromosome 10, between 27 and 35 megabases, we see a QTL segment, and down at the bottom you see the coefficients for allelic identity compared to the Lied scores. The peak of the Lied score shows a suppression by CAST. The CAST allele in this population completely suppressed, not completely, but significantly depressed DNA damage, showing it to be a very strong dominant effect in this context. That's supported by the work which shows when you can actually look at all of the genotypes within that group of 150 male mice. You see that on the left, the genotypes associated with CAST in terms of the damage estimated in the bone marrow of micronucleated reticulocytes, you see that they are resistant. All of the other genotypes show varying degrees of susceptibility across this population. Admittedly, there are unequal numbers in this population. The animals were randomly selected, but they were assigned to exposure group by weight, which obviously was not random. Looking again at that QTL segment that was identified, we see that at the bottom, you see the significant variation between CAST and PWD. They're different in these 2 wild-derived lines compared to the other wild-derived line, WSB. All of the inbred lines show no significant variation in this context. They're all identical by descent. Down at the bottom, you see the bar red and black. This is a heat map for the Lied scores. You look down directly beneath the reddest parts of this, you see that there is a candidate gene, NKIN2, also known as TCBA1, as well as a sulfotransferase. This NKIN2 has been associated before with breakpoints in mixed T-cell leukemias, lymphomas. From this point, we can work out through this QTL segment to test and analyze each of these candidate genes to determine their potential significant relationship here. Regressing out the Lied scores for chromosome 10, reveals another significant QTL on chromosome 2 that you can look at the bottom. You see at the intercept of the peak of the Lied score that there appears to be the NZO allele, which actually increases susceptibility. So this is a tool, I believe, which can help us analyze and define the range of variation in terms of susceptibility, and gives us a tool by which to identify the responsible alleles, giving rise to bioinformatics studies and functional validation studies that can show the importance of this. And that's shown here for the QTL. Same thing. If you look at the bottom 3 of the gray and white, the SNP variation for NZO is very different from CAST and PWK. The heat map shows one particular candidate gene. At the center of that, it's a macro D2 gene that has been associated in GY studies in humans associated with a number of different diseases. If you filter that data and those 2 different QTL segments by the amount of variation within those mice, with those genotypes, you see that there are sets of candidate genes within that peak that show significant genetic variation. We can use that to seed knowledge-based pathways. Going back, this is not nearly as sophisticated as what Dr. Schott showed us, but it shows us that we can look through the hand-curated literature, looking at binary interactions between the proposed candidate genes, shown here in red circles, that can look for, as I said, these binary interactions that can be used to seed hub-centric networks that point out that task space 1 in the panel to the top left, which shows some significance, shows hub-centric networks associated with beta-catenin networks. In the lower, both C-SARC and cyclin D1, and at the top, less significant were the networks that are associated with probably P53 networks. But this will allow us to use this data and from this discovery phase in a hypothesis for hypothesis-based research. So therefore the value of population-based models, I think, has been shown. We've shown that both ADME can be a heritable quantitative trait, that benzene-induced DNA damage appears to be a heritable trait. And we can use this data from this population showing this range of variation to stratify them into quartiles and calculate the benchmark dose for each of the different ranges of susceptibility in this context. And we've done that, but I don't have time I can't show you that today. So by taking a genetically diverse population of mice, a limited number, I remind you, we can perform statistical analysis for these genotype-phenotype associations and identify candidates for causal mechanisms and use those for validation through hypothesis-based research testing. Most importantly, I started out telling you about the collaborative cross mice, these 300 lines of recombinant inbreds, which actually can be used for functional validation. You can match the relevant genotypes in the collaborative cross lines to those with either resistant or susceptible genotypes in the DO mice and perform functional validation and even create either stem cells or IPS lines to look at in vitro as well as in vivo targeted testing mechanisms. My time's almost up. This is a very collaborative project designed and conceived at the NTP with a number of individuals playing important roles. The ADME studies were directed by Mike Cunningham. They were conducted at the University of Arizona in Glenn Sipes' lab. Gabe Knudsen recently got his PhD from this work. The flow cytometry work was done at ILS. Lion conducted the inhalation studies, and the linkage analysis was done by Dan Gaddy and Gary Churchill at Jackson. And I owe a debt of gratitude to David Threadgill and Fernando Pardo Manuel de Villena, who have helped me understand the significance of this work. Thank you.
That was a great presentation. Really interested in the results you're generating. I wanted to just respond with a comment that you made at the beginning of your talk about the Land article in Science. Steve Rappaport and I and Martin Smith are part of that group. You had said that the paper wasn't able to detect a no effect exposure level, or wasn't able to detect it.
Right.
Well, that study wasn't designed to detect it. The question that was posed by that study was...
It makes a good story.
Right. That's true. But I'll finish. Let me continue just for a second. So the study was designed to ask the question: is there evidence of metatoxicity at relatively low effects in occupational setting? And what was striking about our work is that, in fact, there was no threshold. There are some people in the world who believe that there's a threshold with regard to benzene's carcinogenicity and hematotoxicity. With a bit of a dearth, we believe, of experimental and epidemiologic evidence supporting that view. in our study, we found no level. And in fact, additional work that Steve has done suggests that there may be evidence of, there may be increased risk, reason to suspect increased risk at very low levels, at environmental levels of exposure. The other point I wanted to raise is that, so our study is of 240 workers. They're all Han Chinese from the same part of China. Probably more diversity than your inbred mice who aren't exactly genetically identical. But still, we've identified genetic factors that seem to modify benzene hematotoxicity. We've just completed doing a genome-wide scan, so we're doing QTL analysis on the same work you're doing. But we see effects, as far as the main effect of benzene hematotoxicity, well below 1 part per million. In your models, you seem to have to go up to 100 parts per million to see effects. So although on one hand we're really interested in comparing the results of the main hits you have with the results that we have in humans who are exposed to benzene, I do wonder about the generalizability of your findings potentially because, as Steve has shown, there are different enzymes that play different roles at different levels of exposure. 100 parts, fortunately, hopefully, nobody in the world is exposed to 100 parts per million benzene now, though they were not all that long ago. But there may be some developing countries where levels can get that high. But given that we see effects well below 1 ppm and your models don't seem to show that, I just wonder about the generalizability of your findings.
Well, for the sake of time, I didn't give you some information. And there is a significant trend in a dose response. So both 1 and 10 and 100 show a significant trend. 100 against air is highly significant. 10 against air is highly significant. But 1 was not. But within that population of 1, there were significant individuals which showed increased DNA damage. I've not shown you the hematotoxicity data. Had to be done by hand, and it's still being analyzed. So you made a very important. Those are very important points. I don't know how generalizable we can make it across that because I don't know exactly how this population of mice might correlate with the Han Chinese population that you observed. And I expect we chose 1 simply because that was the exposure level that we could control under environmental conditions. They don't have the luxury that you have with having Steve.
Finally, we're looking forward to comparing results, as it may actually help to validate your, not that there's a gold standard, but if in fact we see the same underlying changes in the same genes, that would be striking and would actually support.
That's true. Well, that's going to be very interesting to find out how you can compare in the candidate genes from that strength of association when you're looking genome-wide and where the different size effects are. Because I know they play a role, but the QTL was so dramatic. The lot score was over 14.5. It's a significant dominant effect. I just don't know how yet it relates to human exposure.
Thanks, Jeff.